All right, welcome everybody to Biogeography. Like I said, we'll have a short break in the middle because we had a couple cancellations. Eric, you can go ahead and get started.
Okay, thank you for being here. I will speak about my part of my PhD thesis. I titled this presentation like Environmental and Geographic Drivers of Cryptic Diversity of Porites panamensis from the Gulf of California. So first of all, I'm going to talk about scleractinian corals. Scleractinian corals are the reef builders of coral reef that are one of the most important ecosystem in the sea. But when we start to have genome data, which that there are a lot of colonies that are morphological with have sensation. Evidence of uh, they have evidence of barriers to gene flow. So we call these colonies like cryptic diversity. So cryptic diversity can be found across ranges of spatial scales from colonies only a meter apart. For example, here this is a coral reef. We can observe cryptic diversity that are in sympatric. For example, in one area of the coral reef, and we can observe another colony that are in the lagoon and another in the slope. But also we can observe cryptic diversity across hundreds or thousands of kilometers. For example, in this image, we can observe in cryptic diversity in temperature gradients or light gradients. I will focus on this scenario because here in the Gulf of California, we have scleractinian corals, but in the Gulf of California exhibit environmental heterogeneity. For example, this is a seascape classification. In the northern region, we can observe seasonality of surface temperature, also high neutron levels and turbidity levels driven by whaling events. And in the southern region here, in the southern Gulf of California, we can observe water masses warmer and with clearer conditions than the northern. Finally, here in the middle of Gulf of California, this is a transitional zone between the temperate waters in the north and tropical waters at the south. Also, the speed and direction of the ocean currents depends on the season. This heterogeneity has been linked with genetic structure observed in other marine biodiversity. For example, in this red-head goblet, they observed two populations, one in the north and another in the south, but with outlier loci. Those loci that its variation is driven by selection, putting selection. They observed another group here that in purple one that was in the middle of the Gulf of California that matched with the transitional zone in the Gulf inside the basin. So this also they perform random forest and they observed that the outlier this pattern of genetic diversity was also explained by temperature ranges. So this temperature could drive to put adaptive scenarios in marine populations here in the Gulf of California. So going back to scleractinian corals, this is Porites panamensis. This is a picture. Porites panamensis is one of the most important reef builders here in the eastern tropical Pacific. This is a map of its distribution. But the interesting thing about this species is that it's a brooder. So it releases its larvae in a developed stage. So we can expect a lot of auto recruitment between colonies that are close. Also, it tolerates low temperature and high turbidity because it exhibits a lot of phenotypic and physiological plasticity in response to the environmental gradient that they have in its distribution. Something interesting is, according to the niche model, the center of abundance of Porites panamensis is in the north of the Gulf of California. And with ribosomal DNA sequencing, they observed that one population is here inside the Gulf of California with high levels of gene flow. However, we know with genome data that we can observe cryptic diversity, and we have here in the Gulf of California a lot of heterogeneity. For that reason, we want to answer how do the geographic and the environmental heterogeneity of the Gulf of California shape the spatial patterns of genetic diversity in Porites panamensis? So to answer this question, we sampled in 19 sites inside the Gulf of California. We covered most of the area. We collected around 140 samples of adult colonies of Porites panamensis. I performed the direct sequencing of these samples. I trimmed the bad quality reads. I trimmed the reads that were mapped with the symbiont, and I kept just the remaining reads, and then those reads were mapped with the Porites panamensis reference genome that was recently sequenced. I performed the snip calling in stacks. I performed a lot of filterings, and in order to identify SNPs that are under neutral evolution and under putative selection evolution or outlier loci, I performed three genome scans. In order to identify genetic differentiation, we use the neutral loci, the insulation by distance, and genetic flow also work with the neutral SNPs. And SNPs outlier were used with redundance analysis in order to test if the environmental heterogeneity can drive to one part of the genetic diversity of Porites panamensis using temperature, turbidity, and current velocity as a predictors for the redundance analysis. After the filtering, I kept 2,447 SNPs and of 135 colonies from the Gulf of California. With the genome scans, I identified just 14 SNPs as outliers, and the remaining reads were neutral. In this case, were 2,153 SNPs. With those neutral loci, we identify high genetic structure among populations in the PCA. This is the PCA. Each circle is the colony, and the color represent the sample site. So the first thing that we observed in the PCA was that in San Carlos Bay, that are in the continental side, that is here in the northernmost, the San Carlos Bay is here in the continental side, was separated from the rest, but also the northernmost colonies in Puerto Peñasco, that is here in the continental side, were also differentiated. So on the continental coast of the Gulf of California, two genetically distinct groups were identified. In the case on the peninsula side, that is here, we observe at the mouth of the Gulf of California another group, especially in the Frailes and Los Cabos. The remaining colonies from from Ayala Pass to the Midriff Island were Anmites. We observed this pattern too in the structural analysis. This is according to Evanotes, the best partition is three groups. Again, the group at the south of Gulf of California was differentiated. The one in the continental side in San Carlos was differentiated, and another group with high admixing individuals. But when we test with KF4, we observed that the Puerto Peñasco population was separated. That is the northernmost colonies that are here in the Gulf of California. According to the genetic differentiation analysis with EFT comparison between colonies, we observed that again, in the southern part of the Gulf of California, the colonies were separated from the rest, and the northernmost side was separated from the rest. So we have a mantle test. We perform a mantle test in order to test if there are insulation by distance. In the X ads, we have the genetic distances, Euclidean distances, and in the white ads is the genetic differentiation, and we observe a correlation. So a part of the genetic diversity or structure that we observe here is explained by the distance. But also, when we observe the deep analysis in order to identify the magnitude and direction of gene flow, we observe that we have a lot of gene flow among colonies that are in the peninsula side. Just the colonies that are in the south are insulated. this pattern could be because here in the peninsula side, we have a lot of rocky reef. There's a K factor for sediment and act as a stepping stones for larval dispersal. Also here in the peninsula side during the summer, that is the reproductive peak of the Porites panamensis, we observe that currents are bidirectional, so can disperse larvae through sites here in the peninsula side. In the other part, in the case of the population that we observe in the continental side, and is more complex, probably something related with the demography history. So, but I didn't analyze this part. So I'm working in that. In the case of the outlier loci, we observed that all the descriptors were I can use the descriptors in the model because we're significant, and we can observe the colonies from the south are related with the temperature and the population that are in San Carlos Bay in the continental side. Probably the turbidity is shaping this pattern. So probably Porites panamensis might be differently adapted to distinct environments inside the Gulf of California. So in summary, a strong genetic structure was observed for Porites panamensis. At mites, individuals were distributed for the southern peninsula and the Madeira Island, indicating gene flow among colonies. These spatial genetic patterns are consistent with the influence association of the currents and localized substrate availability for larval dispersion and sediment dynamics. And also the redundance analysis revealed significant signals of putative local adaptation, suggesting that Porites panamensis may be differently adapted to the distinct environment conditions across the Gulf. Thanks for all people that helped me a lot during the development of this project. Muchas gracias.
Great, thank you. We have a couple of minutes for questions. You can go ahead and raise your hand or put something in the Q&A. I don't think I see any hands. I have a quick question. Do you expect that you would see similar patterns in other species, or do you think it's specific to the way that these corals disperse?
I think it's the way of the coral disperse, but I believe that this pattern is something related with historical demography patterns because there are a lot of refuge zones here inside the Gulf of California, especially the base. And so I think it's another part that I need to answer in the future, in the coming days.
Cool. There is someone asked in the Q&A, could you go back 2 slides? I don't know if there was a follow-up there, but maybe they wanted to see a map or something.
Okay. Sorry, I stopped to sharing. Sorry, I didn't.
It's okay, we've got a little bit of time.
Yes, here. Any question?
Well, we've got another one in here. It says, interesting. I wonder if the GEA RDA outliers showed association with any specific environmental variables, question mark. Maybe I missed.
Okay. So yes, they suppose that turbidity and surface temperature was significant in the model. So we expect that these patterns could drive part of the genetic diversity observed.
All right, thanks. I think we'll go ahead, let you stop sharing. We can get Nick's slides up there. We have
Okay.
about 30 seconds before we'll get started.
Thank you.
I've got one more question in the Q&A, but I'll just put that in the chat and you can go ahead and answer that if you would like to in the chat.
Quick audio test.
We can hear you.
Okay, great. Thank you.
All right, I'll let you know with just a few more seconds.
Coming
All right,
down.
you can go ahead and get started.
Okay. Thank you for joining. My talk is going to be on the evolution of aquatic plants and specifically trying to investigate the timing and spatial distribution of aquatic plant origins. So aquatic plants, if you're not familiar, they have a similar evolutionary trajectory to certain groups of animals where there were terrestrial plants, and many of those terrestrial plants, after ancestrally evolving to be on land, subsequently got into the water. And so what we call aquatic plants are secondarily aquatic. They're vascular plants, and a lot of times they've lost some of their support structures to get back into the water. So I've got a photo here of aquatic animal and an aquatic plant, depending on where you're coming from. Lots of them distributed across the phylogeny of the flowering plants or the angiosperms. Lots of independent origins, clear convergent evolution, and advantage to getting into the aquatic habitat for various reasons. Also, there's a really wide range of adaptive strategies, adaptive histories, and sort of a spectrum of whether we have these groups that are largely aquatic or entirely aquatic, and then other ones that are only partially aquatic. So some of these are the two groups I have shown on the slide here. The top row are an order called Elismatales that are all considered to be aquatic. The bottom as well, the water lily order Nymphaeales, those are aquatic as well. They're ancient, fully aquatic lineages. Other cases, we have lots of recent origins. So kind of going along with that convergent evolution, not surprising that lots of plants over various times have figured out how to become aquatic and take advantage of the aquatic habitat. So I've shown here 3 columns now, and I have an aquatic species on the top and a terrestrial, or at least a wetland species on the bottom. One of the more striking examples is on the left there in the genus Crassula. This you might know as the jade plant or some of these succulent desert plants. The same genus has this aquatic plant, Crassula aquatica, and that would be a relatively rare instance in that genus of otherwise mostly dry desert plants. So that's really part of the challenge of this project is if we could simply define aquatic plants in simple terms, this might be a little easier, but we have a range of adaptations and a range of maybe cutoffs where people would differently think that plants are aquatic or not quite qualifying as aquatic. So I've got some examples on the screen here. Some plants that are aquatic would have no debate about them being aquatic. They live fully in the water. Some of them pollinate underwater. So those are obviously aquatic plants, but on the right hand of the image here are corn plants or maize. So this plant that people wouldn't suspect of being aquatic can tolerate wet soils and for brief periods of time. So clearly there's some cutoff here where you would say that at some point they're no longer aquatic. I think we could probably agree that maize plants are not aquatic, but if you see them in water and they survive a flood, brief flood, then they're still kind of in the water habitat. So this is kind of the difficulty of making the cutoff and deciding who is aquatic and who's not. From an evolutionary standpoint, the evolution into aquatic habitats is basically a one-way trip. It involves a lot of loss of the structures that would have helped plants to survive out of water. So things like cuticle that prevent plants from drying out, support tissues. A lot of times those are lost, especially for these very aquatic plants. And shown here are, this is a strategy for dealing with aquatic weeds, is just to take them out of the water and dry them out. And there's no plants really going to recover from that in this aquatic community. Okay, so this project kind of came about because this is a really opportune time to investigate something like this. There have been previous efforts to basically document which plants are aquatic. These days we're in the era of big data, and so we have lists of plants that are aquatic. So fortunately, I can kind of springboard off of other people's studies where they have listed the plants. That top study there is from 1999, and that was a kind of early phylogenetic perspective on the same sort of thing I'm trying to do. Obviously, in the last 25 years, there's been a whole lot of advances in phylogeny data. So our understanding of phylogenetic relationships, our ability to build phylogenies. So it's sort of putting these two things together, a list of plants and modern phylogenies, and trying to figure out if there's some patterns that would be meaningful. Reason to suspect a pattern is maybe that there were global changes in environment in certain places, and you could say maybe some environments favored aquatic plants at different times in the past. Shown at the bottom here, just the kind of the distribution, taxonomic distribution of aquatic plants. So they're widespread broadly across many, about half of the orders of aquatic plants. But as you go toward the right of these pie charts, there are fewer and fewer. If you look within taxonomic groups such as a genus, often very few genera have aquatic members, even though about half of orders have aquatic members. So they're around, but not terribly abundant. We're looking at about 1% of flowering plants being aquatic. So I don't know if that's all showing up. Two big questions: how many times have flowering plants evolved to be aquatic, and are there any patterns that go along with that, spatial or temporal? So I'm going to show you a couple of images like this. So this is a phylogeny. Basically, I reconstructed new phylogenies as much as I was able using available data, and put onto that a biogeography estimation. That's the colors that go along with the map there. So the global, these are ecoregions, so 6 ecoregions of the world, very broad scale. And then the pie charts are a reconstruction of where the ancestors were reconstructed to have grown. So you can see changes in location, whether those maybe correlate with plants becoming aquatic. The white rings on there, white ring means that that species is aquatic, and then heading back in time, that would be a reconstruction of an aquatic ancestor. So it's a sort of pie chart on top of a pie chart. The background is aquatic, and the foreground is the geography. Okay, so like I said, I'm taking advantage of lots of other people's data. Very grateful for that. Other advances are, as I mentioned, just the phylogeny that we have. There's also a time-calibrated phylogeny for flowering plants that I can take advantage of. I was hoping just to sort of put a time on these aquatic plant origins using the phylogenies. I guess you can read that to come back to it later if you want to. In putting this together, ran into a few challenges. So one challenge is that the data that people post, there's a ton of data out there, and DNA sequence data sometimes are incorrectly identified. Sometimes that's very apparent. So I've got an example here that's really undeniable. In the raspberry genus, rubus is a sequence. This is a sequence alignment. You can tell that it very clearly doesn't match its neighbors. And if you extract that and do a blast search, you find that it goes to a whole different order of plants. So for whatever reason, I think there's a question sometimes of proper identification of specimens. There are potentials for contamination. This is my little plug for making sure that you verify your data, double check, take a critical approach, and make sure that you're publishing data that are reasonable for the species you think you're publishing. Other issues could be that the cleaning up of data after sequencing might not be thorough enough. And so basically, I tried to take a more critical perspective as far as seeing if these were genuine differences or not. Okay, another one is that not every species has a comprehensive set of DNA sequence data. You can get artifacts like this, where if 2 species in the same genus have sequence representation from different genes, then you might not actually be able to compare them, even though you have DNA data for both of them. So sometimes you'll see this pattern where 2 species in the same genus that really should be monophyletic are not coming up at the same part of the tree. Trying to build a bunch of trees for all of flowering plants was maybe a big ambitious step for this project. Okay, so let's get some results here. So origins, as far as where these aquatic plant origins were located, roughly evenly distributed across the 6 ecoregions, maybe kind of skewed for relative area of these ecoregions. So Australasia coming up a little bit smaller. I reconstructed 612, give or take, which is over twice as many as were previously estimated. I'll get into that a little bit. So the evidence for these being independent would be on a phylogeny. The ancestral reconstructions for those white aquatic charts they're not coming up with a common ancestor. So we have in this genus Grotiola 1, 2, 3 origins, and the aquatic species of Grotile are not monophyletic, and so we have to infer 3 origins to become aquatic for those. Other cases down here, we've got lots of aquatic species in a clade that the ancestor is coming up mostly aquatic. This becomes sort of evidence to investigate in kind of take a second look at some of these species and say, well, maybe we should qualify this as aquatic. So this genus Dynostoma pulled a photo of this plant in water, but it wasn't on the list of plants that are aquatic. So if we made that aquatic, we would have a whole clade of everybody being aquatic and maybe a little cleaner reconstruction. It sort of highlights the challenge of getting these just right. Okay, so overall, these origins are very recent, within the last 10 million years. These are all the origins. What I was maybe hoping to find with this project was a clustering in the deeper past, but pretty even number of these throughout time, and then a whole bunch in the recent. That's consistent across all the different ecoregions. We see an exponential increase in lineages, whether you're looking at terrestrial or aquatic lineages. This obviously goes along with just having a phylogeny. With the dichotomous branching of a phylogeny, you get more and more lineages exponentially toward the present. But there's a little bit of a pattern where the aquatics have a skew toward more recent than even the terrestrial, also illustrated here. There's one way to look at this. This is z-score. So these are bins where the terrestrial plants would have had an equal number of nodes at any one of these points. And so skew toward more points or more origins in the aquatics than expected would be higher. The number below 0 would be fewer than expected, kind of expecting that even number across. And you see there's a pretty big uptick at the very last couple million years there. And that's also consistent across ecoregions. I think this might, it's kind of up in the air. It might be an artifact of how we define these aquatic plants. So having lots of independent origins at the present could be that we're allowing only a few of these species to be aquatic, and maybe more of them really should be considered aquatic by this definition. This is an example. The genus Persicaria are commonly lots of wetland plants. And so where do you draw the line of a wetland plant or an aquatic plant? Another way to look at that is you could also define these more narrowly. So you could be more inclusive or less inclusive, and either one of those would give you fewer origins as a reconstruction. So I think this is a lot of what we're seeing with that many origins at the present. Some of these are evidence for potentially reevaluating the native status of some species. So this is a species that is related to a lot of North American species, but itself is found much more widespread in the world. Might be some worth reevaluating whether that's as native as they say in these green areas on the map. Okay, so recent origins, chance to reevaluate how we define aquatic plants. No big pattern, what I set out to find, but I think we're making a big step forward in basically reconstructing aquatic plant evolution in lots of different contexts. All right, so all the photos, very appreciative of the folks who take those and put them up online and then just let me use them with an attribution. Very fun project to work on, and I would be happy to take any questions.
Great, thank you. We actually have a couple withdraws for the next 2 sessions, so we have a little extra time for questions. Okay. So feel free to put them in the Q&A or raise your hand. I have a couple small questions. Oh, let's go with Sarah, go ahead.
Great talk. Thank you for sharing. My question is, have you thought about taking a life history situated approach and taken into consideration, for example, proportions of the life cycle spent in water, or maybe the types of pollinators or type of pollination involved in these various origin points?
Great question. Thanks. Not explicitly yet. It's a little bit of a work in progress. Actually, I was hopeful that as part of this meeting, I'd get inspired to pursue new questions. So that'll go on my list now. Offhand, I would say that they're distributed pretty evenly. There's within the qualification of whether a plant is aquatic or not, there are subcategories, and so the fully underwater plants are defined differently than plants with floating leaves. So yeah, one of my goals is maybe to take a pass where we trim out these plants that are maybe a little questionable as to how aquatic they are. These plants that one of the things I didn't mention, the way that plants are categorized by the USDA in the United States, they get a wetland classification, and that is measured by basically how well or how often you might find them growing out of water. And so mostly, I think maybe all of these are in this obligate wetland category, but even wetland is sort of not necessarily in the water, could be in wet soils, and so it's a lot of gray area. That I think what I'm hoping to do, I think, with this is open the door and say, well, these need more study, these are well understood, and kind of give other people maybe a chance to pick up where I've set up a question.
Lydia, you can unmute yourself.
Thank you. Super cool talk. Have you noticed any kind of like patterns of like sort of sequential transitions from sort of one strategy to a fully aquatic strategy? Like, do you find in all of these aquatic species that they're kind of closely related to, or even sister clades with things that are wetland or wet soil? Or have you also found situations where there seems to be just like a full-blown transition into a completely aquatic lifestyle without any kind of sort of, you know, prerequisite of an intermediate?
Right. Yes. So they're definitely both kinds. I showed you that example of basically the desert plants that have a one-off aquatic plant. The Persicaria genus, as I mentioned, has a lot of wetland plants that are maybe you could easily be convinced to fall on one side or the other of being aquatic. So I think there's sort of groups that are known to be wetland, at least wetland plants, and then how aquatic they are is a little bit up for debate. Let's see, back here, too far back. So this one in the middle, Bacopa, I did a project on that genus previously. The one I'm showing here is, it's got these dissected leaves where they've lost a lot of their leaf blade tissue, and that's an adaptation for growing in the water. That's only, I believe, the only place you find this plant growing. And nonetheless, it's in a genus that just, when we talk about the genus, we say this is a wetland genus. So I think we can, if we're looking to reconstruct these origins in a morphological, ecological way, it makes, I'd say there are more cases of plants that are already a little wetland adapted becoming more aquatic, and fewer cases of a plant just going all in all at once to become aquatic, and its relatives are not very aquatic.
All right, I think we've got another hand up. Lang.
Thank you so much. This was a great talk. So I'm just curious to know what kind of methodology or just method you use in the biogeographic analysis.
I'm sorry, what kind of what?
what kind of method or you used in biogeographical analysis?
Right. I use the Biogeobears
Uh-huh.
method. So each species got one or more ecoregion where it currently grows and is considered native. So this didn't consider plants having an invasive or adventive range. So parts of the world where it's considered to be native, and then ancestrally would just be the chance of those ancestors being in the same place or having moved from a different place. In my experience, it tends to be pretty obvious. A lot of times, you have a clade that's in a different place than so that the smaller clade of Gratiola species among a big clade of a bunch of them that are in North America. We don't really need a reconstruction to tell us that there's going to be a shift in location somewhere along there. But it does help to have just have kind of an impartial analysis to do that for us.
Okay. Thank you.
Thanks.
Christopher, you can go ahead and unmute yourself.
Can you hear me?
Yes.
Excellent. Thank you so much. That was a super interesting talk. First, very quick question, followed by a second longer one. One, how do you feel having 30 minutes for Q&A?
I was wondering how it was going to go. I didn't have, I'm good with it.
Okay, yeah. It's nice. It allows us to talk about this a little bit more. And then, so I guess my question was, I like to think about seed dispersal a ton. And I was thinking about how that would fit into the story that you were telling. And I guess my main question was, because I usually, I think about seed dispersal in terrestrial habitats, but like, are there any kinds of dispersal modes that might predispose a plant to evolving to become aquatic? And like, what does that look like? Because I imagine the way that plants disperse aquatically, I know that it's different than terrestrially, but how does that fit in with the story that you're telling?
Right. So I guess one thing comes to mind that there are not a lot of fleshy fruits or sort of animal enticing seed dispersals in aquatic plants. Aquatics, they have sometimes adaptations for like adhering to the outside of a duck, for example, or passing through the gut of a animal that's eating aquatic vegetation. So I guess you could maybe see that as a pre-adaptation that helps is to have some kind of seed that I guess takes advantage of the movement of animals around water bodies, or let's see, I guess we don't like also wind dispersal for seeds, not too common. I guess you could stretch. Sometimes you have like an emergent wetland plant that's got wind dispersal. So I guess I don't know offhand. There's a lot of kind of generic seeds that don't have an apparent dispersal adaptation, and I'm also a little curious always about how seeds get around, and I don't study it per se, and so it's always a little wondrous to me how you make a million seeds and they get where they need to go, or at least some of them do. And so yeah, I guess I'd be open to learning more about that, but I don't have great insights on that right now.
Thanks.
I have a super quick one about the data. So I use a lot of open source data too, so I feel that pain. And I'm curious if you noticed that there was a lot of variation across the different taxonomic groups and like how messy or clean the data was, because I see that in animals a lot. It's like different in different groups. I'm just curious if it's similar in plants.
I after running into a few that were misidentified, after a little while, I started making a list of them, and they're pretty evenly distributed across the taxonomy. There will be, a group of them that show up from the same study. Sometimes get a little suspicious about the data source from that perspective. with the abundance of new data technique, data gathering techniques, there are, I would say, some labs that are putting out data where they're not maybe primarily concerned about proper identification. They might be doing high volume with maybe low taxonomic expertise, and so that sometimes comes out. But I try not to. I don't want to point fingers. There's enough of them distributed across lots of different studies that I'd say it's just for a person using someone else's data. You've got to sort of convince yourself, but with less evidence. So if you get sequence data out of your own lab, you can look at the source. You maybe have your own sources of contamination. You can evaluate the data quality. When you get sequences from GenBank, it's just nucleotide letters. And so you have to kind of infer what I was showing about maybe they had low quality chromatograms or the misidentified specimens. You have to sort of infer that. And I think there's probably a ton that are lost in the middle of like minimally misidentified or minimal errors. The ones I showed were egregious. The ones that it's in the completely wrong plant group, and if you do a blast search, it's a 98% match to something very far away. So yeah, I think ideally, and I think we're moving that way, that eventually we're just gonna be drowning in so many sequence data that we'll probably overcome this. But it's also at an early stage, having a sequence to go along with a species is an important first step, and we got to get that first step right. So yeah. People are building on top of that, and maybe they would use your wrong data to misidentify the next sequence or something.
Yeah, I agree. Hopefully going forward, people just think a little bit more about it. We had a couple things in the Q&A. One person asked, can I get your email address, please? I'd like to ask more questions. Looking at the slides again, it's a lot of info to digest in such a short time slot. So I don't know if you want to share that in the chat. If I hit type answer, Sarah, can he answer that? I don't know how that works.
Yes. And also that question is actually directed at Erick, if he's still here.
Oh, Erick, I'm sorry.
I don't know.
Good call.
Yeah, so if Erick wants to, he could hit type answer and respond. And I saw that he did that earlier.
Okay.
So he could go ahead and do that.
And then there was one other in the Q&A comment. Some aquatic plants do not bear seeds and propagate clonally.
Right. That's a good point. I skipped over that when answering the seed question. Yeah, absolutely. That's among plants, the aquatic plants are some of the best at getting around vegetatively. So fragmentation, I mentioned, sticking to parts of waterfowl. You can do that also with bits of leaf that also will move a plant from one place to another. That's a question for a whole other project, though.
All right, great. Thank you so much.
Yeah,
so we'll have about a 15-minute break before we get to our next talk. Okay, and I'll let you know when it's 12:15 exactly.
Okay. Good afternoon, everyone. My name is Mingming. I'm a postdoc from Laval University from Quebec, Canada. Thank you for joining me today. I'm happy to share with you my study, Genomic Insights into Invasion History and Adaptive Evolution in an Invasive Insect, the Asian Longhorn Beetle. Invasive species are those not native to a region and can cause harm to the environment, the economy, and even human health within that region. They can include plants, animals, fungi, or microbiomes. It is the second biggest threat to biodiversity following habitat loss. For example, the invasive plants Myconia can destroy native habitats by preventing sunlight from reaching the plants beneath it. Besides ecological damage, invasive species also impose massive economic costs. Another concern with biological invasion is that with globalization, there's no signs of a slowdown in the introduction of invasive species. This means we are constantly facing new challenges related to biological invasions. In general, the invasion process is composed of 4 stages: transport, introduction, establishment, and the spread. At each of these stages, actions can be taken accordingly, from preventative measures to chemical, physical, or biological control. Among all these strategies, early detection and eradication are the most cost-effective, such as visual surveys, bait traps, and biosurveillance animals. Nowadays, genomic methods offer promising tools for identifying invasive species in an early stage. For example, DNA barcoding can rapidly determine a specimen's identity. By comparing the genomes of native and invasive populations, we can reconstruct the invasion pathways, revealing where it's from and how it's spread. We can also identify genomic signatures associated with traits that leads to its invasion success. Now, my focal species, the Asian longhorn beetle, it feeds on various tree species such as maples, poplar, and willow. It is one of the most damaging invasive species, and its larvae exhibit excellent cold tolerance ability, which contributes to its invasion success. It is native to China and South Korea and was first introduced into New York in 1996 through wooden packaging material and has since spread across North America and Europe. Using genomic methods, I'm interested in finding out whether the populations are genetically distinct, and I aim to identify the source regions of these populations, knowing whether they originate from a single native region or from multiple sources. And finally, I'm also investigating whether there's evidence of local adaptation to temperature across the species' native range. First, let's look at only the native populations, as a clear-cut population delimitation is necessary for identifying the source of invasion. We collected samples across its native range, including 20 locations spanning 4 major geographic regions in China, namely the North, Northeast, Northwest, and the South. We sequenced the genomes of these native populations using genotyping by sequencing technology. After SNP calling and filtering, the dataset was refined to 6,000 SNPs. We examined population structure using multiple methods. From the results, these figures summarize the native population structure. In the left figure, which is called a procrust analysis, we can simply see it as a twist of principal components aligned with its geographic coordinates. This shows that the geographically close populations exhibit more similar genetic makeup. On the red panel, it's the admixture plot. Each tiny bar represents an individual, and the color composition in each bar shows the ancestry distribution, meaning that the more similar the color composition, the closer the genetic relatedness. We can see that the population across the native range can be divided into several genetic groups. Now let's move on to the invasive populations. We collected invasive samples from 5 states in the US and from Toronto, Canada, and we reused the native samples. In this study, to facilitate the assignment of invasive populations, we divided the native populations into 5 groups based on our previous findings. The sequencing and data pre-processing followed the same procedure as described before. So to begin with, let's look at the phylogenetic tree, including all samples. The gray branches represent native samples, whereas the colored ones represent invasive samples. We see that individuals from certain regions form single, well-supported branches. For example, you can see those highlighted in light gray chunks. and some invasive samples are nested within them. This provides evidence that those invasive populations originated from specific native regions. There are also invasive samples that did not nest within any native branches, such as the New Jersey, New York, and Illinois samples in the middle of the tree. These samples also show mixed genetic composition, as shown in the bar plot, suggesting that they originate from more than one ancestral population. Next, I used the DAPC assignment method to assign invasive individuals to the 5 native genetic groups. As expected, samples from New Jersey and New York, most from New York, were assigned to various regions, as they showed complex genetic composition, as we've seen before. Combining all previous findings, we already kind of have a rough idea of which invasive populations originate from which native regions. And we can hypothesize several invasion scenarios. And by using DIY ABC, we can test these scenarios and estimate parameters for inferring invasion history. In our analysis, the New Jersey and most New York samples were excluded because previous studies have suggested that simpler models yield more accurate estimations in this analysis. So the results from this analysis tell us how and when population divergence occurred, the duration of genetic bottlenecks, and changes in effective population sizes. And our findings indicate that a specific region, the North 2 region, located in the Middle East of China, is the source of most North American invasions. Last but not least, we also examined local adaptation to temperature factors. According to a previous study, it found that temperature accounts for most ALB occurrences in China using spatial analysis, highlighting the important role of temperature in ALB occurrence. Here, I aim to use genomics to explain how climate factors contribute to local adaptation in ALB. First, using genomic data alone, we performed the selection scans to identify outliers putatively under selection. By integrating genomic data with environmental information, we can identify genetic signatures specifically linked to certain variables. In this study, we employed a pooled DNA sequencing strategy. We selected 10 groups based on genetic similarity and the local temperature, and also avoiding relatedness. For each group, DNAs from 20 individuals were equally mixed together, and we performed the whole genome sequencing using NovaSeq. After SNP calling and cleaning, we retained 4 million clean SNPs. And the temperature variables selected were 2 uncorrelated bioclimatic indices, mean temperature, coldest month, and the isothermality. In summary, this study involved 3 categories of variables: the geographic distance, the genetic distance, and the environmental distance between populations, totaling to 7 matrices. And I calculated the correlation coefficients among these 7 matrices. The results are shown in this network figure. The solid lines represent significant correlations. The darker the color, the higher the coefficient value. We see that the mean temperature of coldest month is strongly correlated with all datasets, suggesting a strong impact of temperature on the genetic variation of ALB populations. By associating temperature variables with genetic markers, we were able to identify genes most strongly associated with temperature adaptation. We identified approximately 2,000 significant genes combining both methods. Among the top hits, we found several genes and proteins known to contribute to insect co-tolerance, such as cryoprotectants, which are key agents that help insects overwinter. We identified several significant cryoprotectant-related genes, such as glycerokinase, trilose transporter, and ice structure protein. Additionally, we identified multiple heat shock proteins, which is a stress-induced gene family, commonly found upregulated during cold or heat stress in insects. All the significant genes were enriched in 90 KEGG pathways. Many of these pathways are known to be involved in co-tolerance processes. For example, diabols. This is the most studied process in insects coping with low temperature. And this was well represented in our results. And many known diabols-related pathways were enriched in our analysis, as shown here on the list. So the large number of significant genes suggests that the temperature adaptation is a complex process involving a broad set of genes. With our analysis, we quantify and dissect which genes and pathways are most involved in the process. And to conclude, we examine native populations and detect genetically distinct groups within the native range, and we identified the source populations of North American invasions and inferred multiple independent introduction events and reconstructed their invasion history. Finally, by associating genetic and environmental variables, we identified genes involved in temperature-driven local adaptation, confirming established knowledge while also providing new insights. So thanks to my team and all the partners, I would be happy to take questions.
Great, thank you so much. We have a couple minutes for questions, so feel free to raise your hand or put something in the Q&A. Xinfa, go ahead and unmute yourself.
All right, thank you. Interesting research. I mean, first, I was just wondering how this type of research could add to the xenophobic bias or discrimination against people of Asian descent, given the current political environment. But I would like to point out that the invasive species, it goes in many different directions. So there has been invasion across the continent in different directions. But related to your talk, I was just wondering, when in one slide you showed the phylogenetic tree or network that has the years on them. Yeah, the one you just passed. The other one. Yes, here. So I was just wondering why there was a dash line and where those years came from.
Okay. About your concern about the xenophobic, so previously there are already some population genetic studies using microsatellite markers to trying to review the invasion pathways globally. So it's within ALB is quite well known the global invasion pathways is not only from China to North America. There's also spread within the invasive range, for example, between Europe and North America. So, but my data set doesn't cover the European samples, so I couldn't infer that. So yeah, at least within the Asian longhorn beetle, it's well known that it's not only from Asia to like other corner of the world. It's kind of like everywhere. It's spreading everywhere. So this study, we are hoping to help preventing like further spread between continents. So focusing on this specific analysis, the dashed lines represent the bottleneck events. Bottleneck, so I'm not sure if the audience is well versed with the population genetics. So bottleneck is a common process during invasion, during invasion. So in the beginning of the invasion, the population size remains small. And following the invasion success, the population gradually expand. So in the model setting, for the invasive populations, I manually set up the bottleneck events for each invasion. And those years, first, we give prior knowledges to this model based on previous literatures and according to the biologies of this species. And eventually, it estimates the posterior parameters. And of course, it doesn't directly tell us, oh, it's from 1996 to 2010. It's based on, there's some date conversion manually to give us such precise years.
I'm sorry, it's the year based on the other type of document.
Yeah. I did not so this paper has been published, so all the supplemental figures and tables is public. So I can share the citation, and if you're interested, you can go into that supplemental table. It clearly tells how these years were inferred.
So do you see, I'm sorry.
Yeah, we're kind of out of
Kunjulakshmi.
time here. People are going to want to go take some lunch breaks. But maybe you guys can hook up if you want to chat about that more.
Sure. Yeah, thanks.
Great. Thank you so much. Thank you, Mingming, and all the other